Deep inside every cell of your body, a molecule so ancient and so powerful holds the blueprint to everything you are, everything you were, and everything you could ever become. It is the architect of your eye color, the author of your heartbeat, the silent engineer behind every breath you have ever taken. It is called deoxyribonucleic acid. You know it simply as DNA, and what you think you know about it barely scratches the surface. Tonight, we are going to take you on a journey into the most astonishing, bizarre, and mind-bending truths about the molecule that makes life possible. Some of these facts will shock you. Some will make you rethink everything you believed about biology, and some will leave you staring at your own hands, wondering how something so impossibly complex could exist inside something as ordinary as a skin cell. Please let us know in the comments where you are watching this video and what time you are watching it. And be sure to subscribe to the channel for future videos. Now, let us begin. If you could take the DNA from just one of your cells and stretch it out into a single thin line, it would extend to roughly 6 feet long. That is taller than most human beings, but here is where it gets truly staggering. Your body contains approximately 37 trillion cells. If you were to uncoil every strand of DNA from every single cell in your body and lay them end to end, the total length would stretch to roughly twice the diameter of the solar system. We are talking about a distance that could reach from the sun all the way past Pluto and back again. All of that genetic material, packed inside a body that stands just a few feet tall. The compression ratio is almost incomprehensible. Your cells manage to fold, twist, and condense nearly 6 feet of molecular thread into a nucleus that is only about 6 micrometers across. That is smaller than a speck of dust. Every single cell in your body performs this extraordinary feat of packaging, and it does so without tangling, without breaking, and without error most of the time. Think about that for a moment. You are walking around right now carrying a molecular thread long enough to leave the solar system, folded into a space so small you cannot see it with the naked eye. And your body assembled all of it from scratch, every strand, every coil, every twist. Now consider this. Every single day, your body produces roughly 3.8 million new cells every single second. That means your cellular machinery is copying, duplicating, and packaging 6 feet of DNA billions of times each day. The speed at which your DNA replication machinery operates is extraordinary. The enzyme responsible for copying your DNA, called DNA polymerase, can add approximately 1,000 nucleotides per second in human cells. And the remarkable thing is not just the speed, it is the accuracy. DNA polymerase makes roughly 1 error for every 100,000 nucleotides it copies. But your cells have built-in proofreading mechanisms that catch and correct the vast majority of those mistakes, reducing the final error rate to roughly 1 mistake per billion nucleotides. That is the equivalent of typing out the entire contents of a vast library and making only a single typo across all of those millions of pages. But even that astonishing accuracy is not perfect, and those rare, uncorrected errors are what we call mutations. Mutations are the raw material of evolution, the tiny imperfections that over millions of years have given rise to every species that has ever lived on this planet. Without those 1 in a billion mistakes, life as we know it would never have diversified beyond the simplest single-celled organisms. The very mechanism that keeps your DNA faithful is also the mechanism that allows life to change, adapt, and evolve. Here is something that most people never stop to consider. You share approximately 99.9% of your DNA with every other human being on the planet. Every person you have ever met, every stranger you have passed on the street, every historical figure you have ever read about, you share nearly all of your genetic code with them. The entire spectrum of human diversity, every difference in height, skin color, facial structure, predisposition to disease, personality tendency, and physical ability, all of that variation comes from just 0.1% of your genome. That sliver of genetic difference is what makes you uniquely you, and yet it is almost nothing compared to what you share with the rest of humanity. But the surprises do not stop at humans. Your DNA connects you to every living organism on the planet in ways that most people find deeply unsettling. You share approximately 60% of your DNA with a banana. Yes, the fruit, the yellow thing you put in your cereal. 60% of the genetic instructions that build a banana are also present in your genome. You share roughly 70% of your DNA with a sea slug. You share about 85% of your DNA with a mouse. And you share a staggering 98.7% of your DNA with chimpanzees. The genetic difference between you and a chimpanzee is smaller than the genetic difference between a horse and a zebra. From a purely molecular standpoint, you are almost indistinguishable from the great apes. What this reveals is that life on Earth shares a common ancestor. The fundamental genetic toolkit, the genes that control basic cellular functions, energy production, cell division, and protein construction, those genes have been conserved across hundreds of millions of years of evolution. Nature does not reinvent the wheel. When evolution finds a gene that works, it keeps it. And so the genes that help a yeast cell metabolize sugar are remarkably similar to the genes that help your own cells do the same thing. The code is ancient. The code is shared. And the code connects you to every bacterium, every plant, every insect, and every whale that has ever lived. Let us talk about what DNA actually looks like, because the popular image most people carry in their minds is not entirely accurate. You have probably seen the double helix, two spiraling strands wound around each other like a twisted ladder. That image, first described by James Watson and Francis Crick in 1953, is one of the most iconic images in all of science. But what most people do not realize is that Watson and Crick did not actually discover the structure of DNA on their own. Their model was built upon critical experimental work performed by Rosalind Franklin, a brilliant crystallographer whose X-ray diffraction images of DNA provided the key data that made the double helix model possible. Franklin's famous photograph 51, an X-ray image she captured in 1952, clearly showed the helical structure of DNA and provided measurements that were essential to building an accurate model. Watson and Crick used Franklin's data, controversially, without her explicit permission or knowledge at the time, to construct their model. Franklin died of ovarian cancer in 1958 at the age of 37, likely due to extensive exposure to X-ray radiation during her research. She never received the Nobel Prize. Watson, Crick, and their colleague Maurice Wilkins received the Nobel Prize in Physiology or Medicine in 1962. Franklin's contribution was largely unacknowledged for decades. Today, she is recognized as one of the most important scientists in the history of molecular biology, and her story serves as a sobering reminder of how scientific credit has not always been distributed fairly. Now, the double helix that Watson and Crick described is actually just one form of DNA. It is called bDNA, and it is the form most commonly found in living cells under normal physiological conditions. But DNA can also adopt other configurations. A DNA is a shorter, wider right-handed helix that forms under dehydrated conditions. zDNA is a left-handed helix. It spirals in the opposite direction from bDNA, and it has been found in certain regions of the genome where it may play a role in gene regulation. There are even more exotic forms, including triplex DNA and quadruplex DNA, which involve 3 or 4 strands instead of 2. Quadruplexes, which form in guanine-rich sequences, have been found at the ends of chromosomes and in the promoter regions of certain genes, including some genes associated with cancer. The idea that DNA is always a simple double helix is a simplification. The molecule is far more structurally versatile than most people realize. Let us talk about the building blocks of DNA. The entire genetic code is written using just 4 chemical letters: adenine, thymine, cytosine, and guanine, commonly abbreviated as A, T, C, and G. That is it. 4 letters, the entirety of biological complexity on Earth. Every flower, every fungus, every fish, every human being is encoded using a 4-letter alphabet. Your genome contains roughly 3.2 billion of these letters, arranged in a precise sequence that constitutes your genetic blueprint. If you were to print out your entire DNA sequence in standard text, it would fill roughly 262,000 pages. That is a stack of books taller than a person, and all of that information is stored inside a nucleus the size of a pinpoint. The way these 4 letters encode information is through groups of 3, called codons. Each codon specifies a particular amino acid, the building blocks of proteins. There are 64 possible 3-letter combinations of A, T, C, and G, but there are only 20 standard amino acids. This means the genetic code is redundant. Multiple codons can code for the same amino acid. This redundancy is not a flaw, it is a buffer. It means that some mutations in the 3rd position of a codon have no effect on the protein that gets made. These are called silent mutations, and they provide a layer of protection against the damage that random genetic errors might otherwise cause. Now, here's something that will surprise many people. Only about 1.5% of your DNA actually codes for proteins. That means the vast majority of your genome, roughly 98.5%, does not contain instructions for building proteins. For decades, scientists referred to this non-coding DNA as junk DNA, a term that implied it served no purpose. It was thought to be the accumulated debris of millions of years of evolution, broken genes, viral remnants, repetitive sequences that had lost their function. But that label turned out to be profoundly wrong. The Encyclopedia of DNA Elements project, known as ENCODE, published landmark findings in 2012 demonstrating that approximately 80% of the human genome has some form of biochemical function. Much of the so-called junk DNA is involved in regulating when and where genes are turned on and off. These regulatory regions act like switches, dimmers, and volume knobs, controlling the activity of genes with extraordinary precision. Without these regulatory sequences, your cells would not know when to become a heart cell versus a liver cell versus a neuron. The junk turned out to be the control panel. Some of the most fascinating non-coding DNA consists of remnants of ancient viruses. Roughly 8% of your genome is made up of sequences that originated from retroviruses, viruses that inserted their genetic material into the DNA of your ancestors millions of years ago. These sequences are called endogenous retroviruses, and most of them are now inactive, but some of them have been co-opted by your body and repurposed for essential functions. One of the most striking examples involves a gene called syncytin. Syncytin is derived from an ancient retroviral gene, and it plays a critical role in the formation of the placenta. Without this repurposed viral gene, mammalian pregnancy, as we know it, might not be possible. Your ability to develop inside your mother's womb is in part thanks to a virus that infected one of your distant ancestors tens of millions of years ago. Evolution did not just tolerate this viral invasion, it weaponized it. Let us move deeper into the world of your genome. Your DNA is organized into structures called chromosomes. Humans typically have 46 chromosomes arranged in 23 pairs. You received 1 chromosome in each pair from your mother and 1 from your father. These chromosomes are numbered roughly by size, with chromosome 1 being the largest and chromosome 22 being one of the smallest. The 23rd pair consists of the sex chromosomes, XX in females and XY in males. But here is a remarkable detail that most people do not know. The Y chromosome, which determines male biological development, is the smallest human chromosome and contains only about 55 genes. The X chromosome, by contrast, contains roughly 800 genes. Over evolutionary time, the Y chromosome has been steadily shrinking. Millions of years ago, the X and Y chromosomes were roughly the same size. But because the Y chromosome does not recombine with another Y during reproduction, it is always passed from father to son without a matching partner. It has gradually accumulated mutations and lost genes. Some geneticists have speculated that the Y chromosome could eventually disappear entirely, though this is a subject of active debate and would not occur for millions of years if it happens at all. The ends of your chromosomes are capped by structures called telomeres. Telomeres are repetitive sequences of DNA that do not code for any protein. Their job is purely protective. They function like the plastic tips on the ends of shoelaces, preventing the chromosome from fraying or fusing with neighboring chromosomes. But here is the critical detail. Every time a cell divides and copies its DNA, the telomeres get a little bit shorter. The DNA replication machinery cannot fully copy the very end of a linear chromosome, so a small portion of the telomere is lost with each cell division. Over time, after many divisions, the telomeres become critically short, and the cell enters a state called senescence. It stops dividing and eventually dies. This process is one of the fundamental mechanisms of aging. The shortening of your telomeres is, in a very real sense, a molecular clock counting down the lifespan of your cells. There is an enzyme called telomerase that can rebuild telomeres and counteract this shortening. Telomerase is highly active in certain cells, stem cells, reproductive cells, and immune cells that need to divide many times without aging out. But in most of your body's cells, telomerase is turned off or expressed at very low levels. This is actually a protective mechanism, because cells that can divide indefinitely without telomere shortening have a much higher risk of becoming cancerous. In fact, roughly 90% of human cancers have reactivated telomerase, allowing the cancer cells to divide without limit. The very mechanism that could theoretically prevent aging is also the mechanism that enables cancer. Biology, it turns out, is full of cruel trade-offs. Speaking of cancer, let us talk about how your DNA protects itself from damage. Every single day, your DNA sustains between 10,000 and 100,000 individual lesions per cell. These can be caused by ultraviolet radiation from the sun, reactive oxygen species generated by your own metabolism, chemical exposure, and simple random errors. If all of this damage went unrepaired, your genome would quickly become non-functional. But your cells possess an astonishingly sophisticated suite of DNA repair mechanisms. There are at least 5 major repair pathways, each specialized for different types of damage. Base excision repair fixes small chemical alterations to individual bases. Nucleotide excision repair removes bulky lesions caused by UV light. Mismatch repair corrects errors introduced during DNA replication. Homologous recombination and non-homologous end joining repair double-strand breaks, the most dangerous type of DNA damage, in which both strands of the double helix are severed. These repair systems work around the clock, constantly scanning your DNA, detecting errors, and fixing them before they can cause harm. When these repair systems fail or are overwhelmed, the result can be cancer, premature aging, or genetic disease. One of the most well-known examples of a DNA repair deficiency is a condition called xeroderma pigmentosum. People with this rare genetic disorder have a defective nucleotide excision repair pathway, which means their cells cannot properly repair the DNA damage caused by ultraviolet light. As a result, even brief exposure to sunlight causes severe burns and dramatically increases the risk of skin cancer. People with xeroderma pigmentosum must avoid sunlight entirely and live primarily indoors or at night. They are sometimes called children of the moon. It is a heartbreaking condition that illustrates just how essential DNA repair is to survival. Let us now consider something almost philosophical. When we say that DNA contains the instructions for building a living organism, what do we really mean? The analogy most people use is that DNA is like a blueprint, but that analogy is incomplete. A blueprint for a building shows exactly where every wall, window, and door should go. DNA does not work that way. DNA does not contain a map of where every cell in your body should be. Instead, it contains instructions for making proteins, along with elaborate regulatory information about when and where those proteins should be produced. The body builds itself through a cascade of chemical signals, feedback loops, and environmental interactions that unfold over time. It is less like following a blueprint and more like executing a recipe where the ingredients themselves react to each other and modify the process as it proceeds. The development of a human being from a single fertilized egg is not a simple read out of genetic instructions. It is an enormously complex, dynamic process in which genes interact with each other, and with the environment in ways that scientists are still struggling to fully understand. Here is an example that illustrates this beautifully. Identical twins share 100% of their DNA. They develop from the same fertilized egg, which splits into two embryos early in development. And yet, identical twins are not perfectly identical. They may differ in height, weight, personality, disease susceptibility, and even fingerprints. Their fingerprints are always different because fingerprint patterns are influenced by the exact conditions in the womb: the pressure, the position, the flow of amniotic fluid during a critical window of fetal development. DNA sets the broad parameters, but the environment fills in the details. This phenomenon is part of what scientists call epigenetics, the study of how gene expression is modified by factors outside the DNA sequence itself. Epigenetics is one of the most revolutionary frontiers in modern biology. The term literally means above genetics, and it refers to chemical modifications that sit on top of DNA and influence which genes are active and which are silent without changing the underlying sequence. The most well-studied epigenetic modification is DNA methylation, in which a small chemical group called a methyl group is added to the cytosine base in DNA. When a gene's promoter region is heavily methylated, that gene is typically silenced, turned off. When the methyl groups are removed, the gene can be activated. This is how a liver cell and a brain cell can contain exactly the same DNA but behave in radically different ways. They have different patterns of methylation that activate different sets of genes. What makes epigenetics truly astonishing is that some epigenetic modifications can be influenced by the environment, by diet, stress, toxin exposure, and life experience, and some of these modifications can be passed from one generation to the next. This means that the experiences of your parents and even your grandparents may have left chemical marks on your DNA that influence your biology today. Studies in animal models have shown that mice exposed to certain stressors can pass altered stress responses to their offspring, even when those offspring are raised in completely stress-free environments. The implications for human health are profound and still being explored. Your genes are not your destiny. They are a starting point, a canvas upon which life paints its modifications. Now let us talk about a piece of DNA that you carry, but that does not come from both of your parents. Inside nearly every cell of your body, there are small, bean-shaped structures called mitochondria. Mitochondria are the powerhouses of the cell, generating the energy currency known as ATP that fuels nearly all of your biological processes. But mitochondria have their own DNA, a small, circular chromosome that is entirely separate from the 46 chromosomes in the cell nucleus. Human mitochondrial DNA contains just 37 genes, compared to the roughly 20,000 protein-coding genes in the nuclear genome. And here's the crucial detail: mitochondrial DNA is inherited exclusively from your mother. When a sperm fertilizes an egg, the sperm contributes almost no mitochondria to the resulting embryo. The mitochondria in your cells are descended from the mitochondria in your mother's egg cell, which were descended from the mitochondria in her mother's egg cell, and so on, stretching back through an unbroken maternal line for hundreds of thousands of years. This purely maternal inheritance pattern has given scientists a powerful tool for tracing human ancestry. By analyzing mutations that have accumulated in mitochondrial DNA over thousands of generations, researchers have been able to reconstruct the migration patterns of human populations across the globe. This work led to the concept of mitochondrial Eve, the most recent common matrilineal ancestor of all living humans. Mitochondrial Eve is estimated to have lived roughly 200,000 years ago in Africa. This does not mean she was the only woman alive at that time. It means that she is the woman from whom all living humans have inherited their mitochondrial DNA through an unbroken chain of mothers. Every person alive today carries mitochondrial DNA that traces back to this single woman. It is a humbling reminder that no matter how diverse humanity appears on the surface, we are all connected by a single maternal lineage. There is a parallel concept on the paternal side. The Y chromosome, because it is passed only from father to son, can be used to trace paternal lineages in much the same way. Y chromosomal atom is the most recent common patrilineal ancestor of all living humans, estimated to have lived between 200,000 and 300,000 years ago, also in Africa. Interestingly, mitochondrial Eve and Y chromosomal atom almost certainly did not live at the same time or in the same place. They are simply the endpoints of two different genealogical tracking methods. Let us now delve into something that sounds like science fiction but is scientific fact. Your body is not entirely human. From a genetic standpoint, you are a chimera, a composite organism made up of both human and non-human DNA. The human microbiome, the collection of bacteria, viruses, fungi, and other microorganisms that live in and on your body, contains an enormous amount of genetic material. Recent estimates suggest that the number of microbial cells in your body is roughly equal to the number of human cells, about 30 to 40 trillion. But the genetic diversity of those microbes dwarfs your own. The human genome contains roughly 20,000 protein-coding genes. The combined genomes of the microbes in your gut alone contain somewhere between 2 million and 20 million genes. That means the microbial genes in your body outnumber your own genes by a factor of 100 or more. In a very real genetic sense, you are more microbe than human. These microbial genes are not passive passengers. They perform functions that your own genome cannot. Gut bacteria produce essential vitamins, break down complex carbohydrates that your own enzymes cannot digest, train your immune system to distinguish friend from foe, and even produce neurotransmitters that influence your mood and behavior. The composition of your gut microbiome has been linked to conditions ranging from obesity and diabetes to depression and anxiety. Some researchers now refer to the gut microbiome as a second brain because of its profound influence on neurological function. Your DNA may have built your body, but the DNA of your microbial partners helps run it. Here is another startling fact about the DNA inside you. Not all of the DNA in your cells is doing something useful at any given time. In fact, nearly half of the human genome is composed of transposable elements, sequences of DNA that can move from one location in the genome to another. These mobile genetic elements, sometimes called jumping genes, were first discovered by the geneticist Barbara McClintock in maize plants in the 1940s. Her discovery was met with widespread skepticism because it challenged the prevailing view that genes were fixed in place on chromosomes. It took decades for the scientific community to appreciate the significance of her work, and she was finally awarded the Nobel Prize in Physiology or Medicine in 1983 at the age of 81. Transposable elements come in several varieties. Some, called retrotransposons, copy themselves and paste the copy into a new location, a mechanism similar to how retroviruses replicate. Others, called DNA transposons, cut themselves out of one location and insert themselves into another. The most abundant transposable elements in the human genome are ALU elements, which are short sequences about 300 base pairs long. There are more than 1 million ALU elements in your genome, making up roughly 11% of your total DNA. Most of these elements are no longer active. They have accumulated mutations that have disabled their ability to jump. But some remain capable of transposition, and when they land in the wrong place, they can disrupt gene function and cause disease. ALU insertions have been implicated in cases of hemophilia, breast cancer, and neurofibromatosis, among other conditions. And yet, transposable elements are not merely destructive. Over evolutionary time, some of them have been domesticated by the genome and repurposed for beneficial functions. The RAG proteins that drive the rearrangement of antibody genes in your immune system, the process that allows your body to produce an almost infinite variety of antibodies to fight infection, are derived from an ancient transposable element. Without this co-opted jumping gene, your adaptive immune system would not function. Once again, evolution has taken something potentially dangerous and turned it into something essential. Let us talk about one of the most dramatic examples of DNA's power, the Hox genes. Hox genes are a family of genes that control the basic body plan of an organism during embryonic development. They determine which end of the embryo becomes the head and which end becomes the tail, where the limbs will grow, and how the segments of the body are organized. What is truly remarkable about Hox genes is their deep conservation across the animal kingdom. The Hox genes that pattern the body of a fruit fly are strikingly similar to the Hox genes that pattern the body of a human being. In fact, the order in which Hox genes are arranged on the chromosome mirrors the order in which they are expressed along the body axis, a phenomenon called collinearity. The genes at one end of the cluster control development at the head end of the body, while the genes at the other end control development at the tail end. This organizational principle has been conserved for more than 500 million years. The same genetic logic that tells a fly where to put its wings tells your body where to put your ribs. Mutations in Hox genes can produce dramatic and often bizarre developmental outcomes. In fruit flies, a mutation in the antennae gene causes legs to grow where antennae should be. In another mutation called ultrabithorax, the fly develops an extra pair of wings. These homeotic transformations, where one body part is replaced by another, demonstrate the extraordinary power of Hox genes to direct the fundamental architecture of living organisms. In humans, mutations in Hox genes can cause limb malformations, extra digits, and other developmental anomalies. Now let us consider the human genome from the perspective of information theory. Your genome contains roughly 3.2 billion base pairs. Each base pair can be one of four options, A, T, C, or G, which means each position carries 2 bits of information. That gives the human genome an information content of roughly 6.4 billion bits, or about 800 megabytes. That is roughly the amount of data that fits on a single compact disk. The entirety of what makes you biologically human, every physical trait, every metabolic pathway, every instinct and predisposition, is encoded in less data than a single high-definition movie. When you compare this to the enormous complexity of the human body, the efficiency of DNA as an information storage medium is staggering. In fact, scientists are now exploring the use of DNA as a data storage technology. Researchers have successfully encoded digital information, text, images, video, and even computer programs into synthetic DNA molecules. The advantages of DNA storage are extraordinary. DNA is incredibly dense. A single gram of DNA can theoretically store 215 petabytes of data, that is 215 million gigabytes. DNA is also remarkably durable. Under the right conditions, DNA can remain intact for thousands of years. Researchers have successfully extracted and sequenced DNA from bones that are hundreds of thousands of years old. No hard drive, no flash drive, no magnetic tape can match the longevity and density of DNA as an information storage medium. The molecule that life has been using to store its instructions for billions of years may turn out to be the future of digital data storage as well. Speaking of ancient DNA, let us talk about what we have learned from extracting DNA from long-dead organisms. The field of ancient DNA research has undergone a revolution in the past few decades, thanks to advances in sequencing technology and contamination prevention protocols. Scientists have now extracted and sequenced DNA from organisms that died tens of thousands, even hundreds of thousands of years ago. The oldest DNA ever recovered came from permafrost sediments in Siberia and dates to approximately 2 million years ago. That DNA belonged to ancient plants and animals that lived in an ecosystem completely different from anything that exists in that region today. Perhaps the most famous achievement of ancient DNA research is the sequencing of the Neanderthal genome. Neanderthals were a species of archaic humans that lived in Europe and Western Asia for hundreds of thousands of years before going extinct roughly 40,000 years ago. By extracting DNA from Neanderthal bones found in caves, scientists led by Svante Pääbo, who won the Nobel Prize in Physiology or Medicine in 2022 for this work, were able to reconstruct a nearly complete Neanderthal genome, and what they found was astonishing. Modern humans of European and Asian descent carry between 1 and 4% Neanderthal DNA. This means that when modern humans migrated out of Africa and encountered Neanderthals, the two species interbred. The Neanderthals are not entirely gone. Their DNA lives on inside billions of people alive today. Some of the Neanderthal genes that persist in modern human genomes affect immune function, skin and hair characteristics, and even susceptibility to certain diseases. Some Neanderthal gene variants have been linked to increased risk of depression, blood clotting disorders, and even severe responses to certain infections. But Neanderthals are not the only archaic humans whose DNA survives in modern populations. In 2010, scientists analyzing DNA from a tiny finger bone found in Denisova cave in Siberia discovered an entirely new group of archaic humans, now called the Denisovans. The Denisovans were so different from both modern humans and Neanderthals that they constituted a distinct lineage. And yet, modern human populations in Southeast Asia, Oceania, and parts of East Asia carry Denisovan DNA in some cases, as much as 5 or 6% of their genome. The Denisovan gene variant most famously found in modern humans is a version of the EPAS1 gene, which helps the body cope with low oxygen levels at high altitude. This gene variant is found at high frequency in Tibetan populations and is believed to have been acquired through ancient interbreeding with Denisovans. It is one of the clearest examples of adaptive introgression, a case where DNA from another species provided a survival advantage that was favored by natural selection. Let us now explore something that will change the way you think about inheritance. For most of the history of genetics, scientists believed that the traits you inherit from your parents were determined entirely by the DNA sequence you received. But we now know that this is not the whole story. Epigenetic marks, chemical modifications to DNA and the proteins associated with it, can be passed from parent to child and can influence gene expression across generations without any change to the DNA sequence itself. One of the most striking examples of transgenerational epigenetic inheritance comes from historical studies of famine. Researchers studying the descendants of people who survived extreme famine during the Dutch hunger winter of 1944 to 1945 found that the children and grandchildren of famine survivors had altered metabolic profiles. Grandchildren of women who were pregnant during the famine had higher rates of obesity and cardiovascular disease, even though they themselves had never experienced food scarcity. The most likely explanation is that the famine triggered epigenetic changes in the survivors that were passed down through the germline, the eggs and sperm, to subsequent generations. The starvation of a grandmother left a chemical mark on the DNA that influenced the metabolism of her grandchildren decades later. This finding has profound implications. It suggests that the experiences of your ancestors, what they ate, what they endured, what toxins they were exposed to, may have left epigenetic fingerprints on your genome that influence your health today. You are not just the product of your own genes. You are the product of your ancestors' lives. Now let us talk about a feature of DNA that is both beautiful and essential. The genetic code is nearly universal. With only minor exceptions, every living organism on Earth uses the same genetic code. The codon AUG always means methionine. The codon UGA almost always means stop. From bacteria to blue whales, from mushrooms to maple trees, the same codons specify the same amino acids. This universality is one of the strongest pieces of evidence that all life on Earth shares a common ancestor. At some point in the distant past, roughly 3.5 to 4 billion years ago, a single-celled organism, the last universal common ancestor, sometimes called LUCA, established the genetic code that would be used by every one of its descendants. And every organism alive today is one of those descendants. There are few exceptions to the universality of the genetic code, and they are fascinating in their own right. Some species of bacteria, some mitochondria, and a few single-celled organisms use slightly modified versions of the genetic code in which one or two codons have been reassigned to different amino acids. These variations are extremely rare and are thought to have arisen through evolutionary drift in organisms with very small genomes. But the overwhelming conservation of the genetic code across all of life underscores a fundamental truth. The code was established very early in the history of life and has remained essentially unchanged for nearly 4 billion years. Here is something that may surprise you about genes themselves. Humans do not have significantly more genes than many far simpler organisms. The human genome contains approximately 20,000 to 25,000 protein-coding genes. A fruit fly has about 14,000, a roundworm has about 20,000, a grape plant has about 30,000, more than a human being. The complexity of an organism is not determined simply by the number of genes it possesses. What matters far more is how those genes are regulated, how they interact with each other, and how alternative splicing, a process in which a single gene can produce multiple different proteins by selectively including or excluding different segments of the gene sequence, expands the functional output of the genome. Through alternative splicing, the 20,000 genes in the human genome can produce an estimated 100,000 or more distinct proteins. It is not the number of instruments in the orchestra that matters. It is the complexity of the symphony they play. Let us now discuss an area of DNA science that has transformed medicine, agriculture, law enforcement, and our understanding of ourselves. DNA sequencing, the ability to read the precise order of bases in a DNA molecule, has undergone an astonishing acceleration since the Human Genome Project was completed in 2003. That project, which took 13 years and cost approximately $3 billion, produced the first complete sequence of the human genome. Today, a human genome can be sequenced in less than a day for roughly a few $100s. The cost of sequencing has dropped faster than the cost of computing, faster than Moore's law. This democratization of sequencing technology has opened up possibilities that were unimaginable just 2 decades ago. One of the most immediate applications of cheap, fast DNA sequencing is in personalized medicine. By sequencing a patient's genome, doctors can identify genetic variants that influence how that individual metabolizes drugs, responds to treatments, and is predisposed to disease. Pharmacogenomics, the study of how genes affect a person's response to medication, is already being used to tailor drug prescriptions to individual patients. For example, some people carry a genetic variant that causes them to metabolize the blood thinner warfarin much more slowly than average, making them highly susceptible to dangerous bleeding if given a standard dose. A simple genetic test can identify these individuals and allow doctors to adjust the dose accordingly. This kind of precision medicine, guided by an individual's DNA, is becoming increasingly common and represents a fundamental shift in how healthcare is delivered. DNA sequencing has also revolutionized our understanding of cancer. Cancer is, at its core, a disease of DNA. It arises when mutations accumulate in genes that control cell growth, division, and death. By sequencing the DNA of a patient's tumor and comparing it to the DNA of their healthy cells, scientists can identify the specific mutations driving that individual's cancer. This has led to the development of targeted therapies, drugs designed to attack cancer cells with specific genetic mutations while sparing healthy cells. The drug imatinib, for example, was designed to target a specific genetic abnormality called the BCR-ABL fusion gene, which is found in chronic myeloid leukemia. Before imatinib, the 5-year survival rate for chronic myeloid leukemia was roughly 30%. After imatinib, it rose to approximately 90%. This is the power of understanding cancer at the DNA level. Now, let us talk about one of the most important and controversial technologies in the history of science, CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it is a gene editing technology that allows scientists to make precise changes to the DNA of any living organism. CRISPR was adapted from a natural defense system used by bacteria to fight viral infections. When a bacterium survives a viral attack, it stores a small piece of the virus's DNA in its own genome as a kind of molecular memory. If the same virus attacks again, the bacterium uses that stored sequence to recognize and destroy the viral DNA. Scientists Jennifer Doudna and Emmanuel Charpentier figured out how to repurpose this system as a programmable DNA editing tool, and in 2020, they were awarded the Nobel Prize in Chemistry for their work. CRISPR works by using a guide RNA to direct an enzyme called Cas9 to a specific location in the genome. Once there, Cas9 cuts both strands of the DNA at that precise spot. The cell's natural repair mechanisms then kick in to fix the break, and scientists can manipulate this repair process to delete a gene, correct a mutation, or insert a new piece of DNA. The technology is faster, cheaper, and more accurate than any previous gene editing method, and it works in virtually any organism: bacteria, plants, animals, and human cells. The potential applications of CRISPR are breathtaking. It could be used to cure genetic diseases like sickle cell disease, cystic fibrosis, and muscular dystrophy by correcting the underlying mutations in a patient's DNA. It could be used to engineer crops that are more resistant to drought, pests, and disease. It could be used to eliminate mosquito-borne diseases like malaria by genetically modifying mosquito populations so they can no longer carry the malaria parasite. It could even be used to resurrect extinct species by editing the DNA of closely related living species to match the genome of the extinct one. Scientists are already working on a project to use CRISPR to bring back a version of the woolly mammoth by editing Asian elephant DNA to incorporate mammoth genes for cold tolerance. But CRISPR also raises profound ethical questions. In 2018, a Chinese scientist named He Jiankui announced that he had used CRISPR to edit the genomes of 2 human embryos, which were then carried to term and born as twin girls. He claimed to have disabled a gene called CCR5 in the embryos, with the intention of making the children resistant to HIV infection. The announcement was met with widespread condemnation from the scientific community. The experiment was performed in secret, without proper ethical oversight, and the long-term health consequences for the children are unknown. He Jiankui was sentenced to 3 years in prison by a Chinese court. The incident sparked a global debate about the ethics of human germline editing, modifying DNA in a way that will be inherited by future generations. Most scientists and bioethicists agree that germline editing in humans is not yet safe or ethical, but the technology exists, and the temptation to use it will only grow as the tools become more precise and more accessible. Let us shift our attention now to something more personal. What does your DNA say about where you came from? The consumer genetic testing industry has exploded in recent years, with companies offering to analyze a sample of your saliva and provide a detailed breakdown of your ancestral origins. These tests work by comparing specific genetic markers in your DNA, called single nucleotide polymorphisms, or SNPs, to reference panels of DNA from populations around the world. Because certain genetic variants are more common in certain populations, statisticians can estimate the proportion of your ancestry that traces to different geographic regions. But the results of these tests come with important caveats. The reference panels used by different companies are not identical, which means the same person can get different ancestry estimates from different companies. The estimates are probabilistic, not definitive. And they reflect only a fraction of your total ancestry. You have 2 parents, 4 grandparents, 8 great-grandparents, and so on. Go back just 10 generations, roughly 300 years, and you have over 1,000 ancestors. Go back 20 generations, and you have over 1 million. Not all of those ancestors contributed DNA to you in equal measure, because DNA is inherited in randomly sized chunks. It is entirely possible that some of your ancestors from just 10 or 15 generations ago left no detectable trace in your DNA at all, even though they are unquestionably part of your family tree. DNA ancestry tests tell you about the DNA you inherited, not about all the ancestors you had. Nevertheless, genetic ancestry testing has produced some genuinely surprising and meaningful discoveries. It has reunited adoptees with their biological families. It has solved cold cases and identified the remains of disaster victims. It has overturned family legends and revealed hidden histories, and it has given millions of people a deeper connection to their heritage. Let us talk about DNA fingerprinting and forensic science. The idea that DNA could be used to identify individuals was first demonstrated by the British geneticist Alec Jeffreys in 1984. Jeffreys discovered that certain regions of the human genome contain short sequences that are repeated a variable number of times. The number of repeats at these loci varies from person to person, making each individual's pattern of repeats essentially unique, except in the case of identical twins. Jeffreys called this technique DNA fingerprinting, and it was first used in a legal case in 1986 to identify the perpetrator of 2 murders in Leicestershire, England. The case was groundbreaking. It both exonerated an innocent suspect and led to the conviction of the actual killer. Since then, DNA evidence has become one of the most powerful tools in forensic science. Modern forensic DNA analysis uses a technique called short tandem repeat profiling, which examines the number of repeats at multiple loci across the genome. The probability that 2 unrelated individuals would share the same STR profile across all tested loci is typically less than 1 in a billion, making DNA evidence extraordinarily powerful in court. DNA has been used to convict the guilty, and just as importantly, to exonerate the innocent. Organizations like the Innocence Project have used DNA evidence to free more than 300 wrongfully convicted individuals from prison, some of whom had served decades behind bars for crimes they did not commit. But forensic DNA technology has also raised privacy concerns. Law enforcement agencies have used genealogical databases, including consumer genetic testing databases, to identify suspects through their relatives. The most famous case involved the Golden State Killer, a serial murderer and rapist who had evaded capture for decades. In 2018, investigators uploaded a DNA profile from crime scene evidence to a public genealogical database and identified distant relatives of the suspect. By building a family tree from those matches, they narrowed the search to a single individual, Joseph James DeAngelo, who was subsequently arrested, tried, and convicted. The case was hailed as a triumph of forensic science, but it also raised uncomfortable questions about genetic privacy. When you submit your DNA to a consumer testing service, you are not just sharing your own genetic information. You are sharing information about your parents, your siblings, your children, and your extended family. The genetic data you provide could be used in ways you never anticipated, including in criminal investigations involving relatives you may never have met. Now let us return to the molecule itself and consider one of the most remarkable chemical properties of DNA, its ability to self-replicate. The double-stranded structure of DNA is not just an architectural feature, it is the key to replication. Each strand of the double helix serves as a template for producing a new complementary strand. Because adenine always pairs with thymine and cytosine always pairs with guanine, each strand contains all the information necessary to reconstruct the other. When a cell prepares to divide, the double helix is unwound and separated by an enzyme called helicase. Then DNA polymerase moves along each single strand, reading the sequence and assembling a new complementary strand, base by base. The result is 2 identical copies of the original double-stranded molecule, each consisting of 1 old strand and 1 new strand. This mode of replication is called semi-conservative replication, and it was confirmed experimentally by Matthew Meselson and Franklin Stahl in 1958 in what is often described as the most beautiful experiment in biology. The precision of this process is essential. Without accurate replication, genetic information would degrade rapidly over successive generations of cell division. The fidelity of DNA replication is maintained not only by the selectivity of DNA polymerase, which preferentially inserts the correct complementary base, but also by built-in proofreading functions. DNA polymerase can detect when it has inserted an incorrect base and reverse its direction to remove the error before continuing. After replication, additional mismatch repair enzymes scan the newly synthesized DNA for any remaining errors. The result is a replication system of breathtaking accuracy, one that has allowed genetic information to be transmitted faithfully across billions of years of evolutionary history. There is something poetic about the fact that the molecule responsible for heredity, for passing information from parent to child, from generation to generation, has a structure that inherently facilitates its own copying. The double helix is not just beautiful, it is functionally perfect for the job it performs. Let us talk about one more remarkable aspect of DNA before we conclude this part of our journey. Your DNA is not static. It is not a fixed blueprint that you were born with and will carry unchanged to your grave. Your DNA is constantly being modified, remodeled, and reinterpreted throughout your life. Genes are turned on and off in response to environmental signals. Chemical modifications are added and removed. The three-dimensional structure of your chromosomes changes as different regions of the genome are opened for transcription or closed for silence. Your DNA is alive not in the biological sense, but in the sense that it is a dynamic, responsive, ever-changing information system that adapts to the demands of your life in real time. And here is perhaps the most humbling fact of all. Every atom in the DNA inside your body was forged in a star. The carbon, nitrogen, oxygen, hydrogen, and phosphorus that make up the backbone and bases of your DNA were created in the nuclear furnaces of ancient stars that exploded billions of years ago, scattering their elements across the cosmos. Those elements were incorporated into the dust cloud that formed our solar system, condensed into the planet we call Earth, and were eventually assembled into the first self-replicating molecules that gave rise to life. You are, in the most literal sense, made of stardust, and the molecule that carries the instructions for your existence was built from the ashes of dead stars. That molecule, that ancient, elegant, astonishingly complex thread of chemistry, is inside you right now. It is in every cell of your body. It connects you to your parents, your ancestors, and every living thing on Earth. It is the thread that runs through all of biology, all of evolution, all of life. And we have only just begun to understand it. The story of DNA is the story of life itself. It is a story of replication and mutation, of conservation and innovation, of ancient viruses repurposed and broken genes made whole. It is a story written in a four-letter alphabet and read by molecular machines of extraordinary precision. It is a story that stretches back to the origin of life on Earth and forward to the futures we have not yet imagined. And we are nowhere close to finished telling it. This has been only the first chapter. In the second part of this documentary, we will go even deeper. We will explore the strange world of RNA interference, the bizarre phenomenon of horizontal gene transfer, the unsettling reality of genetic mosaicism, and the awe-inspiring potential of synthetic biology. We will discover how DNA has been found in places no one expected, how genes can be silenced by tiny molecules, and how the genetic code may hold the key to extending human life far beyond its current limits. The code of life has many more secrets to reveal. Welcome back. If you were with us for the first part of this journey, you have already discovered that the DNA inside your body stretches to the edge of the solar system, that you carry ancient viral genes that make pregnancy possible, and that the atoms in your genetic code were forged in the hearts of dying stars. But we have only scratched the surface. The molecule of life has far more secrets to reveal, and some of them will challenge everything you thought you understood about biology, identity, and what it means to be alive. Let us begin this second chapter with a fact that is both disturbing and fascinating. You are not genetically uniform. Most people assume that every cell in their body contains the same DNA, a perfect copy of the genome they inherited at conception. But this assumption is wrong. You are a genetic mosaic. Over the course of your development in your life, mutations have accumulated in individual cells, creating subtle genetic differences between the cells of your body. Your skin cells do not carry exactly the same DNA as your liver cells. Your brain cells do not carry exactly the same DNA as your blood cells. Every time a cell divides, there is a small chance that a new mutation will be introduced, and that mutation will be passed to all of that cell's descendants. Over time, your body becomes a patchwork of genetically distinct cell populations. This phenomenon, called somatic mosaicism, is far more common and far more significant than scientists once believed. Studies have shown that the average human brain contains hundreds or even thousands of somatic mutations that differ from the DNA in the rest of the body. Some of these mutations occur very early in embryonic development, when the embryo consists of just a handful of cells. A mutation that occurs at that stage can end up in a large fraction of the body's tissues, creating a person who is essentially two genetically distinct individuals fused into one. This is not hypothetical. There are documented cases of people who have different blood types in different parts of their body, people whose skin displays patches of different pigmentation due to underlying genetic mosaicism, and people who have been found through genetic testing to be their own twin. One of the most striking examples of mosaicism involves a woman named Lydia Fairchild. In 2002, Fairchild applied for public assistance and was required to undergo DNA testing to prove maternity of her children. The results came back saying she was not the biological mother of her own children she had carried and given birth to. Prosecutors accused her of fraud. It was only when further investigation revealed that Fairchild was a chimera. She had absorbed a twin in the womb, and her reproductive organs carried a different genome than her blood, that the case was resolved. Her ovaries contained DNA from her vanished twin, which meant the children she bore carried DNA that did not match the DNA in her blood cells. Fairchild was, genetically speaking, two people in one body, and she had no idea until a court case nearly tore her family apart. Chimerism, the presence of two or more genetically distinct cell populations in a single organism, can arise through several mechanisms. Twin absorption, as in Fairchild's case, is one. Another is the transfer of cells between a mother and her fetus during pregnancy, a phenomenon known as microchimerism. During pregnancy, fetal cells cross the placenta and enter the mother's bloodstream, where they can persist for decades after the child is born. Researchers have found fetal cells in the brains, hearts, livers, and kidneys of women many years after they gave birth. These cells are genetically distinct from the mother, they carry the father's DNA as well, and they have been found to integrate into the mother's tissues and even participate in tissue repair. The biological consequences of microchimerism are still being studied, but some researchers believe that fetal cells may play a role in maternal immune function, wound healing, and even the development or prevention of certain autoimmune diseases and cancers. A mother literally carries pieces of her children inside her body for the rest of her life. The bond between mother and child is not just emotional, it is cellular, it is genetic. Now let us plunge into a world that overturned one of the most fundamental assumptions in biology. For most of the 20th century, scientists believed that the flow of genetic information was a one-way street. DNA makes RNA, and RNA makes protein. This was called the central dogma of molecular biology, articulated by Francis Crick in 1958. But the central dogma, as it was originally understood, has been challenged and expanded in ways that have revolutionized our understanding of the genome. The first major challenge came from the discovery of reverse transcriptase, an enzyme that copies RNA back into DNA. This enzyme is used by retroviruses like HIV to insert their genetic material into the DNA of the cells they infect. The existence of reverse transcriptase demonstrated that the flow of information from nucleic acid to nucleic acid was not strictly one-directional. RNA could be converted back into DNA, blurring the neat hierarchy of the central dogma. But the more profound revolution came from the discovery that RNA itself is far more than a passive messenger. For decades, RNA was seen primarily as an intermediary, a disposable copy of a gene that served as a temporary template for protein synthesis. We now know that RNA molecules play an enormous range of functional roles in the cell that have nothing to do with encoding proteins. Some RNA molecules act as enzymes, catalyzing chemical reactions just as protein enzymes do. These catalytic RNAs, called ribozymes, were first discovered in the early 1980s by Thomas Sech and Sidney Altman, who shared the Nobel Prize in Chemistry in 1989 for this work. The discovery that RNA could function as both a carrier of genetic information and a catalyst shattered the assumption that proteins were the only biological catalysts and gave rise to the RNA world hypothesis, the idea that life on Earth may have begun with self-replicating RNA molecules before DNA and proteins evolved. One of the most remarkable classes of functional RNA molecules is the family of small interfering RNAs and microRNAs. These tiny RNA molecules, typically only about 20 to 25 nucleotides long, play a critical role in gene regulation through a process called RNA interference, or RNAi. In RNA interference, small RNA molecules bind to complementary sequences in messenger RNA molecules and either trigger their destruction or block their translation into protein. The effect is gene silencing, the targeted shutdown of specific genes without any change to the DNA itself. RNA interference was first described in the roundworm Caenorhabditis elegans by Andrew Fire and Craig Mello, who received the Nobel Prize in Physiology or Medicine in 2006. Since then, RNAi has been found to operate in virtually all multicellular organisms, including humans. Your cells produce hundreds of different microRNAs, each of which can regulate dozens or even hundreds of different target genes. These tiny molecules are master regulators of gene expression, controlling processes as diverse as cell growth, differentiation, immune response, and programmed cell death. Disruptions in microRNA function have been implicated in cancer, heart disease, neurological disorders, and autoimmune conditions. The discovery of RNA interference fundamentally changed our understanding of how genes are regulated and opened up entirely new approaches to treating disease. Scientists are now developing RNA-based therapies that use synthetic small RNAs to silence disease-causing genes. The first RNA drug, called patisiran, was approved for use in 2018 for the treatment of a rare hereditary condition that damages nerves and organs. Let us now explore one of the most alien and unsettling phenomena in the world of genetics: horizontal gene transfer. In the familiar world of animals and plants, genes are passed vertically from parent to offspring. You inherit your genes from your mother and father, they inherited theirs from their parents, and so on in a branching tree of descent. But in the microbial world, genes do not always follow this tidy inheritance pattern. Bacteria can transfer genes directly between individuals that are not parent and offspring, and they can do so across species boundaries. This is horizontal gene transfer, and it is one of the reasons that antibiotic resistance spreads so rapidly through bacterial populations. Bacteria exchange genes through several mechanisms. In transformation, a bacterium picks up free-floating DNA from the environment, often from dead bacteria that have released their genetic material. In transduction, a bacteriophage, a virus that infects bacteria, accidentally packages a piece of bacterial DNA and delivers it to a new host. In conjugation, two bacteria form a physical connection, and one transfers a piece of DNA called a plasmid directly to the other. These mechanisms allow genes for antibiotic resistance, toxin production, and metabolic capabilities to spread horizontally through microbial communities at an alarming rate. A gene that evolves in one species of bacterium can end up in a completely unrelated species within a single generation. What is truly surprising is that horizontal gene transfer is not limited to bacteria. It also occurs in eukaryotes, organisms with complex cells, including animals, plants, and fungi. Researchers have found evidence of horizontal gene transfer from bacteria to insects, from fungi to plants, and even from bacteria to the human genome. One striking example involves the parasitic bacterium Wolbachia, which infects a vast number of insect species. In some cases, entire segments of the Wolbachia genome have been found integrated into the chromosomes of the host insect. These transferred bacterial genes can even be functional, producing proteins in the insect host. The tree of life is not a neatly branching tree at all. It is a tangled web, with genes moving horizontally across branches in ways that complicate our understanding of evolutionary relationships. Now let us turn to a subject that lies at the intersection of genetics and human health. We have already mentioned that only about 1.5% of the human genome codes for proteins, but within that small fraction lies an extraordinary density of information that, when disrupted, can cause devastating disease. There are currently more than 6,000 known single-gene genetic disorders, conditions caused by a mutation in a single gene. These include cystic fibrosis, caused by a mutation in the CFTR gene that disrupts chloride ion transport across cell membranes, leading to thick, sticky mucus in the lungs and digestive system. They include sickle cell disease, caused by a single base change in the beta-globin gene that alters the shape of red blood cells and causes them to clog blood vessels. They include Huntington's disease, caused by an expansion of a repeating CAG sequence in the Huntington gene that produces a toxic protein, leading to progressive neurological degeneration. And they include phenylketonuria, caused by a deficiency in the enzyme that metabolizes the amino acid phenylalanine, leading to intellectual disability if left untreated. The sheer variety of single-gene disorders illustrates the critical importance of precise DNA sequences. In many cases, a change of just one letter out of 3.2 billion is enough to cause life-altering disease. Sickle cell disease, for example, is caused by a single nucleotide change, an A to T substitution in the 6th codon of the beta-globin gene that changes one amino acid from glutamic acid to valine. That single amino acid change causes hemoglobin molecules to polymerize under low oxygen conditions, distorting red blood cells into a rigid sickle shape. One letter. One amino acid, one disease that affects millions of people worldwide. The precision of DNA is both its greatest strength and its greatest vulnerability. But here is a twist that adds an unexpected layer of complexity to the story of genetic disease. The same mutation that causes sickle cell disease also provides a survival advantage against malaria. People who carry one copy of the sickle cell mutation, carriers or heterozygotes, have red blood cells that are inhospitable to the malaria parasite. In regions where malaria is endemic, carriers of the sickle cell trait are more likely to survive to reproductive age than people with two normal copies of the gene. This selective advantage has maintained the sickle cell mutation at high frequencies in populations with a long history of malaria exposure. It is a classic example of balancing selection, a situation where a gene variant is simultaneously harmful in one context and beneficial in another. Evolution does not produce perfect solutions, it produces compromises. A similar story plays out with cystic fibrosis. Carriers of one copy of the CFTR mutation, people who do not have the disease but carry the gene, may have enhanced resistance to cholera and other dehydrating intestinal infections. The altered chloride channel in carriers may reduce the fluid loss caused by these infections, providing a survival advantage in environments where cholera and typhoid were historically common. Once again, a devastating genetic disease persists in the population because carrying one copy of the mutation confers a hidden benefit. Let us now discuss an area of DNA science that is rapidly moving from the laboratory to the clinic: gene therapy. The basic idea behind gene therapy is straightforward. If a disease is caused by a defective gene, why not replace or repair the defective gene? In practice, gene therapy is extraordinarily complex. Delivering a corrected gene to the right cells in the right quantity without triggering an immune response or disrupting other genes has proven to be one of the greatest challenges in modern medicine. The field of gene therapy suffered a devastating setback in 1999 when Jesse Gelsinger, an 18-year-old patient with a mild form of a metabolic disorder called ornithine transcarbamylase deficiency, died during a gene therapy trial at the University of Pennsylvania. Gelsinger's death was caused by a massive immune reaction to the viral vector, the modified virus used to deliver the therapeutic gene. His death led to a temporary halt in gene therapy research and a fundamental reassessment of safety protocols. But the field recovered, and in the 2 decades since, gene therapy has achieved remarkable successes. In 2017, the first gene therapy for a genetic form of blindness, a condition called Leber congenital amaurosis, caused by mutations in the RPE65 gene, was approved for clinical use. The therapy, called Luxturna, delivers a functional copy of the RPE65 gene directly to the retinal cells, restoring vision in patients who would otherwise go blind. In 2019, a gene therapy called Zolgensma was approved for the treatment of spinal muscular atrophy, a devastating neuromuscular disease that is the leading genetic cause of death in infants. Zolgensma delivers a functional copy of the SMN1 gene and has been shown to dramatically improve motor function and survival in treated children. These therapies represent the fulfillment of a promise that was first made decades ago, that understanding the genetic basis of disease would lead to cures that address the root cause rather than merely managing symptoms. And now, with CRISPR technology advancing at a breathtaking pace, the next generation of gene therapies is on the horizon. In December 2023, the first CRISPR-based therapy was approved for the treatment of sickle cell disease. The therapy, called Casgevy, works by editing a patient's own blood stem cells to reactivate the production of fetal hemoglobin, a form of hemoglobin that is normally produced during fetal development and is gradually replaced by adult hemoglobin after birth. Fetal hemoglobin does not contain the sickle cell mutation and can compensate for the defective adult hemoglobin. Early results have been remarkable, with treated patients experiencing dramatic reductions in pain crises and hospitalizations. For a disease that has caused suffering for millennia, the arrival of a gene-editing cure represents a watershed moment in human medicine. Let us now venture into one of the most philosophically provocative frontiers of DNA science, synthetic biology. Synthetic biology aims to design and construct new biological systems or redesign existing ones from the ground up. And in 2010, a team led by Craig Venter achieved something that had never been done before. They created a living cell controlled entirely by a synthetic genome. The team synthesized the complete genome of a small bacterium called Mycoplasma mycoides, over 1 million base pairs of DNA, using chemical synthesis, and then inserted this synthetic genome into a recipient cell that had its own DNA removed. The recipient cell, now running on the artificial genome, was able to grow and divide normally. It was, in a meaningful sense, the first organism whose genetic instructions were written entirely by humans. Venter's synthetic cell was a proof of concept, a demonstration that life could be booted up from a chemically synthesized set of instructions. Since then, synthetic biology has continued to push boundaries. Scientists have created bacteria with expanded genetic codes, organisms that use synthetic base pairs beyond the natural A, T, C, and G. In 2014, Floyd Romsberg and his team at the Scripps Research Institute created a bacterium that incorporated 2 unnatural base pairs, dubbed X and Y, into its DNA alongside the 4 natural bases. This semi-synthetic organism represented the first life form with a 6-letter genetic alphabet. While the organism relied on the unnatural bases being supplied from outside, it demonstrated that the genetic code is not immutable. It can be expanded, rewritten, and redesigned. Researchers have also created minimal genomes, stripped-down versions of natural genomes that contain only the genes essential for survival. In 2016, Venter's team created a synthetic organism called JCVI-SYN3-0, which had a genome of only 473 genes, the smallest genome of any independently replicating organism. Remarkably, roughly one-third of those essential genes had unknown functions. Even in the simplest possible living system, there were genes whose purpose scientists could not identify. We understand enough about DNA to build a living cell from scratch, but we still do not fully understand what every gene in that cell does. The humility in that gap between capability and comprehension is one of the defining features of modern biology. Synthetic biology has practical applications that extend far beyond the laboratory. Engineered microorganisms are being used to produce biofuels, pharmaceuticals, and industrial chemicals. Yeast cells have been engineered to produce artemisinic acid, a precursor to the anti-malarial drug artemisinin, which was previously available only from the sweet wormwood plant. This synthetic biology approach has made the drug more affordable and more accessible in regions where malaria kills hundreds of thousands of people each year. Other engineered organisms are being developed to detect environmental pollutants, produce biodegradable plastics, and even manufacture spider silk, one of the strongest natural materials known, in industrial quantities. The DNA of living organisms is being rewritten to serve human purposes in ways that blur the line between biology and engineering. Now let us explore a topic that sounds like pure fantasy but is grounded in rigorous science: de-extinction. Can we bring back species that have vanished from the Earth? The answer, increasingly, is a qualified yes. Using ancient DNA extracted from preserved specimens, combined with modern gene editing techniques, scientists are working to resurrect, or at least approximate, species that went extinct thousands of years ago. The most ambitious de-extinction project currently underway is the effort to bring back the woolly mammoth. A company called Colossal Biosciences, founded in 2021, is working to edit the genome of the Asian elephant, the mammoth's closest living relative, to incorporate mammoth traits such as cold-resistant hemoglobin, thick woolly hair, increased fat reserves, and smaller ears suited to Arctic environments. The goal is not to create a perfect genetic replica of a mammoth, but to produce a cold-adapted elephant that could thrive in Arctic ecosystems, potentially helping to restore degraded tundra habitats. The project is controversial. Critics argue that de-extinction diverts resources from the conservation of currently endangered species, and that the ecological niches once occupied by mammoths no longer exist. Supporters argue that restoring megafauna to degraded ecosystems could have significant environmental benefits, including slowing the thawing of permafrost by compacting snow and maintaining grasslands. The woolly mammoth is not the only candidate for de-extinction. Scientists have discussed the possibility of resurrecting the passenger pigeon, the dodo, the Tasmanian tiger, and even Neanderthals, though the ethical barriers to the latter are obvious and formidable. The technology to rewrite DNA is advancing faster than the ethical frameworks needed to guide its use, and the question of whether we should bring back extinct species is one that society will have to grapple with in the coming decades. Let us now consider what DNA has taught us about the history of life on Earth. By comparing the DNA sequences of different species, scientists can reconstruct the evolutionary relationships between organisms and estimate when different lineages diverged from common ancestors. This approach, called molecular phylogenetics, has produced some startling revelations. For example, molecular evidence has shown that fungi are more closely related to animals than they are to plants. This seems counterintuitive. Mushrooms seem far more plant-like than animal-like, but at the molecular level, the genetic evidence is clear. Animals and fungi share a common ancestor that is more recent than the common ancestor of either group with plants. This means that you are more closely related to a mushroom than a mushroom is to an oak tree. Molecular phylogenetics has also revealed that whales evolved from land-dwelling mammals closely related to modern hippos. The genetic evidence, combined with a stunning fossil record of transitional forms, shows that whales returned to the sea roughly 50 million years ago. Hidden in the DNA of every whale are vestigial genes for legs, hair, and teeth that their land-dwelling ancestors used. Some whale embryos even develop tiny leg buds that are later reabsorbed during development, a ghostly reminder of their terrestrial origins, written in their DNA. DNA has also revealed surprising facts about the pace of evolution. The molecular clock hypothesis, first proposed in the 1960s, suggests that DNA mutations accumulate at a roughly constant rate over time. By calibrating this rate using fossils of known age, scientists can estimate when two species diverged from a common ancestor. Molecular clock analyses have been used to date the divergence of humans and chimpanzees to roughly 6 to 7 million years ago, the divergence of mammals and reptiles to roughly 300 million years ago, and the origin of life itself to roughly 3.5 to 4 billion years ago. These dates, derived from the DNA of living organisms, broadly agree with the fossil record, but have also revealed events that left no fossil trace at all. Let us now talk about one of the most fascinating and least understood features of the genome, non-coding RNA. We have already discussed how only a small percentage of the human genome codes for proteins, but a large fraction of the genome is transcribed into RNA molecules that never get translated into protein. For a long time, these non-coding RNAs were assumed to be biological noise, accidental transcription with no function. But research over the past 2 decades has revealed that non-coding RNAs are critical regulators of cellular function. Long non-coding RNAs, or LNCRNAs, are RNA molecules more than 200 nucleotides in length that do not encode proteins. There are tens of thousands of LNCRNAs in the human genome, and they play diverse roles in gene regulation, chromosome organization, and cell differentiation. One of the most famous LNCRNAs is called XISD. In female mammals, which have 2 X chromosomes, one of the 2 X chromosomes in each cell must be silenced to prevent a double dose of X-linked gene products. XISD is a long non-coding RNA that coats one of the X chromosomes and triggers its inactivation. The silenced X chromosome is condensed into a compact structure called a bar body. This process, called X inactivation, is a remarkable example of how an RNA molecule, not a protein, can control the fate of an entire chromosome. X inactivation also provides one of the most beautiful natural demonstrations of genetic mosaicism. In female cats, the gene for coat color is located on the X chromosome. In heterozygous females, those carrying one allele for orange fur and one allele for black fur, random X inactivation in different cells during development produces patches of orange and black fur, creating the distinctive calico or tortoiseshell pattern. Every calico cat is a living mosaic, a patchwork of genetically distinct cell populations visible to the naked eye. The pattern is determined not by the cat's DNA sequence, but by the random epigenetic silencing of one X chromosome in each cell during embryonic development. Here is a fact about DNA that bridges the gap between biology and computing. Researchers are now developing DNA-based computers, systems that use DNA molecules rather than silicon chips to perform calculations. The idea was first demonstrated by Leonard Adleman in 1994 when he used DNA molecules to solve a mathematical problem known as the Hamiltonian path problem, a problem that involves finding the shortest route through a network of cities. Adleman encoded the cities and routes as DNA sequences and allowed them to react in a test tube. The chemical properties of DNA, specifically, the ability of complementary strands to find and bind to each other, performed the computation automatically. The solution to the problem was identified by analyzing which DNA molecules had assembled correctly. DNA computing is still in its infancy, but it holds extraordinary potential. DNA molecules can perform massively parallel computations, trillions of molecular interactions occurring simultaneously in a single drop of liquid. For certain types of problems, particularly those involving vast numbers of possible combinations, DNA computers could theoretically outperform even the most powerful silicon-based supercomputers. And because DNA computing operates at the molecular scale, it could potentially be used inside living cells, creating programmable biological systems that can sense their environment, perform logical operations, and respond accordingly. Imagine a therapeutic DNA circuit that can detect cancer biomarkers inside a cell, perform a logical computation to confirm the diagnosis, and then trigger the production of a therapeutic protein, all without any external intervention. This is not science fiction. Prototype systems that perform exactly this kind of cellular computation have already been demonstrated in the laboratory. Let us now discuss something that connects DNA to the very origin of life. How did DNA come to exist in the first place? The current scientific consensus is that DNA was not the original genetic material. That distinction almost certainly belongs to RNA. The RNA world hypothesis proposes that early life on Earth was based entirely on RNA, which served as both the genetic material and the catalytic machinery. RNA, unlike DNA, can both store information and catalyze chemical reactions. This dual capability makes it a plausible candidate for the first self-replicating molecule. Over time, DNA evolved to take over the information storage role. It is more chemically stable than RNA, making it better suited for long-term preservation of genetic information, while proteins evolved to take over most of the catalytic functions. RNA was gradually relegated to its current intermediary roles, though it retained its importance in translation, gene regulation, and numerous other processes. The question of how the first RNA molecules arose from non-living chemistry is one of the deepest unsolved problems in science. Researchers have shown that the building blocks of RNA nucleotides can form spontaneously under conditions that may have existed on the early Earth. Laboratory experiments have demonstrated that short RNA molecules can self-assemble, replicate, and even evolve under certain conditions. But the gap between these laboratory demonstrations and a fully self-sustaining RNA-based life form remains significant. How did chemistry become biology? How did a collection of reacting molecules become a living system capable of evolution? These are questions that DNA science alone cannot answer, but they are questions that the study of DNA has brought into sharp focus. Now let us consider the social and ethical dimensions of DNA knowledge. The ability to read, write, and edit DNA has given humanity unprecedented power over the biological world. But with that power comes responsibility, and the history of genetics includes some of the darkest chapters in the history of science. In the early 20th century, the eugenics movement sought to improve the genetic quality of human populations through selective breeding, forced sterilization, and immigration restriction. Eugenics was embraced by governments, universities, and scientific institutions. Tens of thousands of people were forcibly sterilized based on the belief that traits like poverty, mental illness, and criminal behavior were genetically determined and could be eliminated from the population through controlled reproduction. The eugenics movement reached its most horrifying extreme under the Nazi regime, which used eugenic ideology to justify the systematic murder of millions of people deemed genetically unfit. The Holocaust was, in part, a genocide motivated by a perverted understanding of genetics. The legacy of eugenics casts a long shadow over modern genetics. Scientists and ethicists are acutely aware that the tools of genetic analysis can be misused to discriminate, to stigmatize, to reinforce existing social inequalities. As genetic testing becomes more widespread and gene editing technologies become more powerful, the potential for misuse grows. The question of who gets access to genetic technologies, who decides how they are used, and who bears the risks of their application are among the most important ethical questions of our time. Let us shift to a more wondrous aspect of DNA, its role in the diversity of life. The sheer variety of life on Earth, an estimated 8.7 million species, most of which have not yet been scientifically described, is a testament to the creative power of DNA. Every species represents a unique genetic experiment, a distinct solution to the challenges of survival and reproduction. And the diversity of genetic strategies employed by different organisms is staggering. Consider the axolotl, a salamander native to lakes near what is now the center of one of the world's great cities. The axolotl has an extraordinary ability to regenerate lost limbs, organs, and even parts of its brain and spinal cord. This regenerative capacity is encoded in its DNA, and scientists are studying the axolotl's genome, which, at 32 billion base pairs, is roughly 10 times larger than the human genome, to understand the genetic basis of regeneration. If the genes and regulatory networks responsible for axolotl regeneration can be identified and understood, it may one day be possible to activate similar regenerative pathways in human patients, enabling the regrowth of damaged or lost tissues. Consider the tardigrade, a microscopic animal sometimes called a water bear. Tardigrades can survive conditions that would kill virtually any other organism. Extreme heat, extreme cold, intense radiation, the vacuum of outer space, and pressures 6 times greater than those found at the bottom of the deepest ocean. Research on tardigrade DNA has revealed that these organisms possess unique proteins, called damage suppressor proteins or DSUP proteins, that protect their DNA from radiation damage. When researchers inserted the gene for DSUP protein into human cells grown in culture, those cells showed significantly increased resistance to X-ray radiation. The tardigrade's DNA may hold secrets that could one day protect astronauts from the harmful radiation of deep space. Consider the octopus, an invertebrate with a remarkably complex nervous system and sophisticated cognitive abilities. The octopus genome, sequenced in 2015, revealed something astonishing. Octopuses have an extraordinary capacity for RNA editing. While most organisms rarely edit their messenger RNA after transcription, octopuses edit RNA at tens of thousands of sites, effectively recoding the information from their DNA before it is translated into protein. This RNA editing may contribute to the remarkable neural complexity of octopuses, allowing them to fine-tune protein function in ways that DNA mutations alone cannot achieve. The octopus has, in a sense, found a way to expand its genetic repertoire without changing its DNA. Consider the bdelloid rotifers, microscopic freshwater animals that have reproduced entirely without sex for roughly 80 million years. This is almost unprecedented in the animal kingdom, because sexual reproduction is thought to be essential for maintaining genetic diversity and purging harmful mutations. How have bdelloid rotifers survived for so long without sex? Part of the answer appears to lie in their remarkable ability to acquire genes from other organisms through horizontal gene transfer. Bdelloid rotifer genomes contain an unusually high proportion of genes from bacteria, fungi, and plants, genes that appear to have been incorporated into the rotifer genome over millions of years. These foreign genes may provide the genetic diversity that sexual reproduction would normally supply. The rotifers have, in effect, replaced sex with gene theft. Let us talk about one of the most practical and transformative applications of DNA technology: genetic modification of food. Genetically modified organisms, or GMOs, have been part of the food supply for decades. The first commercially available genetically modified food was the flavor-saver tomato, introduced in 1994. Since then, genetically modified crops including corn, soybeans, cotton, and canola have become widespread in global agriculture. These crops have been engineered for a variety of traits, including resistance to insect pests, tolerance to herbicides, enhanced nutritional content, and improved shelf life. One of the most celebrated examples of genetic modification in agriculture is golden rice. Standard rice is a staple food for billions of people, but contains virtually no beta-carotene, the precursor to vitamin A. Vitamin A deficiency is a leading cause of preventable blindness and a major contributor to childhood mortality in the developing world. Golden rice was engineered to produce beta-carotene in the edible part of the grain by introducing genes from maize and a soil bacterium. The result is rice with a golden yellow color that provides a significant source of vitamin A despite its potential to save hundreds of thousands of lives each year. Golden rice has faced decades of regulatory delays and opposition from anti-GMO activists. It was finally approved for commercial cultivation in the Philippines in 2021. The debate over GMOs is complex and multifaceted. The overwhelming scientific consensus is that currently approved genetically modified foods are safe for human consumption, a conclusion supported by thousands of studies and every major scientific organization in the world. But concerns about corporate control of the food supply, environmental impacts, and the potential for unintended consequences remain legitimate topics of discussion. What is undeniable is that the ability to modify the DNA of crop plants has given humanity powerful new tools for addressing food security in a world with a growing population and a changing climate. Let us now return to the human body and explore one of the most paradoxical aspects of DNA, programmed cell death. Your body deliberately kills billions of its own cells every single day. This process, called apoptosis, is essential for health and development. During embryonic development, apoptosis sculpts the fingers and toes by killing the cells between them. Without programmed cell death, humans would have webbed hands and feet. In the immune system, apoptosis eliminates T cells and B cells that might attack the body's own tissues. In adult tissues, apoptosis removes damaged or aging cells to make room for fresh replacements. Apoptosis is controlled by a network of genes that regulate the decision to live or die. When a cell's DNA is damaged beyond repair, tumor suppressor genes like TP53, sometimes called the guardian of the genome, can trigger apoptosis, ensuring that the damaged cell does not continue dividing and potentially become cancerous. The TP53 gene encodes a protein called p53, which monitors the integrity of the cell's DNA and activates repair mechanisms when damage is detected. The damage is too severe, p53 activates the apoptotic pathway, and the cell dismantles itself in an orderly fashion. Mutations that inactivate p53 are found in roughly half of all human cancers. Without a functional p53 protein, damaged cells continue to divide unchecked, accumulating further mutations and progressing toward malignancy. The loss of a single gene, TP53, is one of the most common and consequential events in cancer development. Here is a deeply counterintuitive fact: some organisms appear to barely age at all. The naked mole rat, a small, hairless rodent native to East Africa, has a lifespan of over 30 years, roughly 10 times longer than a similarly sized mouse. Naked mole rats show negligible senescence, meaning they do not exhibit the typical biological deterioration associated with aging. They maintain bone density, muscle mass, and reproductive capacity well into old age. They are also extraordinarily resistant to cancer. In decades of laboratory study, spontaneous cancer in naked mole rats has been documented only a handful of times. Researchers have identified several genetic factors that may contribute to the naked mole rat's exceptional longevity and cancer resistance, including an unusually active version of the p53 gene, enhanced DNA repair mechanisms, and the production of a very high molecular weight form of hyaluronic acid that appears to prevent cells from crowding together and forming tumors. The DNA of the naked mole rat holds clues that could, one day, help extend human health and lifespan. Other organisms challenge our understanding of aging in even more dramatic ways. The immortal jellyfish Turritopsis dohrnii has the ability to revert from its adult form back to its juvenile polyp stage, effectively resetting its biological clock. This process, called transdifferentiation, involves the reprogramming of the organism's differentiated cells back to a more primitive state, a phenomenon that is, at its core, a transformation in gene expression patterns. The jellyfish's DNA does not change during this process. What changes is which genes are turned on and which are turned off. The immortal jellyfish demonstrates that the information for perpetual renewal may already be present in the genome, locked away, waiting for the right epigenetic key to unlock it. Let us explore another astonishing capability of DNA, its use in solving historical mysteries. DNA analysis has been used to identify the remains of historical figures, settle disputes about lineage, and rewrite chapters of history. In 1998, DNA testing confirmed the identity of the remains of Tsar Nicholas II and his family, who were executed by Bolshevik forces in 1918. Mitochondrial DNA extracted from the skeletal remains was compared to DNA from living relatives of the Romanov family, providing a definitive identification. DNA has also been used to study the remains of King Richard III, whose skeleton was discovered beneath a parking lot in Leicester, England, in 2012. Mitochondrial DNA from the skeleton was matched to living descendants of Richard's sister, confirming the identity of the remains. The analysis also revealed that Richard had blue eyes and likely had blonde hair as a child, contradicting some historical depictions. In the world of anthropology, DNA extracted from ancient human remains has transformed our understanding of migration, settlement, and cultural change. The DNA of individuals buried thousands of years ago has revealed that the populations of Europe were almost entirely replaced by waves of migration from the steppes of Central Asia during the Bronze Age. The people who built Stonehenge were largely replaced by newcomers with steppe ancestry within a span of just a few centuries. History, as written in DNA, often tells a story of migration, replacement, and mixing that is far more dynamic and tumultuous than the narrative preserved in archaeological artifacts alone. Now let us turn to a topic that connects DNA to the immune system in one of the most elegant ways imaginable. Your adaptive immune system has the ability to produce antibodies against virtually any foreign molecule it encounters, even synthetic molecules that have never existed in nature. How can a genome with only 20,000 genes produce billions of different antibodies? The answer is one of the most ingenious genetic mechanisms ever discovered, VDJ recombination. The genes encoding antibodies are not stored as fixed, complete sequences. Instead, they are stored as libraries of gene segments, variable, diversity, and joining segments that are randomly shuffled and combined during the development of each immune cell. This process of genetic rearrangement produces a unique antibody gene in each B cell. When you combine the randomness of segment selection with additional mechanisms like junctional diversity, the addition or deletion of random nucleotides at the junctions between segments, and somatic hypermutation, the introduction of targeted mutations into antibody genes during an immune response, the result is an antibody repertoire of almost limitless diversity. Your immune system generates more than 10 billion different antibody specificities, each encoded by a unique DNA sequence assembled on the fly through controlled genetic rearrangement. This is one of the few examples in biology where DNA is deliberately rearranged and mutated as part of normal cellular function. Your immune cells are constantly rewriting their own DNA, shuffling gene segments and introducing mutations at rates thousands of times higher than the background mutation rate. It is controlled chaos, genetic diversity engineered by evolution to protect you against a world of constantly evolving pathogens. Let us discuss an area of DNA research that is still shrouded in mystery: the role of DNA in consciousness and behavior. Is there a genetic basis for intelligence, for personality, for the capacity for language, music, or art? These are some of the most contentious questions in all of science, and the answers are far more nuanced than popular discourse often suggests. Intelligence, as measured by standardized tests, is highly heritable. Twin studies suggest that roughly 50 to 80% of the variation in intelligence between individuals is attributable to genetic factors. But no single gene has been identified that accounts for more than a tiny fraction of this variation. Instead, intelligence appears to be influenced by thousands of genes, each contributing an infinitesimally small effect. The same is true for most complex behavioral traits. Personality, risk-taking behavior, susceptibility to addiction, and even political orientation all show moderate heritability in twin studies, but the specific genes involved are numerous, individually weak in effect, and deeply entangled with environmental factors. The relationship between DNA and behavior is not deterministic. Carrying a genetic variant associated with increased risk of depression does not mean you will become depressed. It means that, all else being equal, your probability of developing depression may be slightly higher than average, but all else is never equal. Your experiences, your relationships, your diet, your sleep patterns, your exposure to stress and trauma, all of these environmental factors interact with your genetic predispositions to produce the person you are. Genes load the gun, as the saying goes, but environment pulls the trigger. And increasingly, scientists are discovering that the relationship between genes and environment is not a simple interaction, but a dynamic, bidirectional feedback loop. Your experiences can change the expression of your genes, and the expression of your genes can influence the experiences you seek out. Nature and nurture are not separate forces. They are intertwined. Let us consider one more breathtaking application of DNA technology before we close. Scientists are now developing gene drives, genetic systems that can spread a modified gene through an entire population of organisms far faster than normal Mendelian inheritance would allow. In normal sexual reproduction, each gene has a 50% chance of being passed to any given offspring. A gene drive increases that probability to nearly 100%, ensuring that the modified gene spreads through the population within just a few generations. Gene drives have been proposed as a tool for combating insect-borne diseases. By releasing mosquitoes carrying a gene drive that renders them incapable of transmitting malaria, it may be possible to eliminate malaria from entire regions within a matter of years. The potential to save hundreds of thousands of lives, mostly children, is enormous. But gene drives also carry significant ecological risks. Once released, a gene drive is difficult or impossible to recall. If the modified gene has unintended effects on the target species or on the ecosystems it inhabits, the consequences could be irreversible. The deliberate modification of a wild species genome is an act of extraordinary hubris and extraordinary responsibility. It is a power that demands the deepest caution and the broadest possible societal deliberation. We have now journeyed through the vast landscape of DNA, from the molecular structure of the double helix to the ethical dilemmas of gene editing, from the ancient viruses buried in your genome to the cutting-edge technologies that may one day cure genetic disease, extend human lifespan, and even resurrect extinct species. We have explored the four-letter alphabet that encodes all of life, the repair mechanisms that protect it, the mutations that diversify it, and the epigenetic modifications that modulate it. We have seen how DNA connects every human being to every other and every species to every other, a single, unbroken thread of molecular information stretching back nearly 4 billion years to the origin of life itself. But perhaps the most important thing to understand about DNA is this: we are still in the early chapters of its story. The human genome was sequenced only 2 decades ago. CRISPR was developed as a gene editing tool barely more than a decade ago. Every year brings new discoveries that overturn old assumptions and open new frontiers. The genome is not a finished book. It is a book we are still learning to read, and one whose language is richer, more subtle, and more layered than we ever imagined. You are made of DNA. Your children are made of DNA. Every living thing on this planet is made of DNA. And the more we understand about this extraordinary molecule, the more we understand about ourselves, our origins, our vulnerabilities, our potential, and our place in the long, unfolding story of life on Earth. The code of life is ancient. The code of life is universal, and the code of life still has secrets left to tell. Thank you for joining us on this journey. The story of DNA is far from over. In many ways, it has only just begun.